Lesson 2.4 is about chemical reactions and the role that enzymes play in those reactions. So at this point, you've already learned about chemical reactions and you know that energy is always involved in a chemical reaction. Living things have to have energy, and so one way that they get it is to either capture it from the sun, or we're going to consume something that did capture energy from the sun and store it in these biological molecules. Another concept that you come to understand is the idea of activation energy. So you understand now that it takes a certain amount of extra energy in order to get a chemical reaction to begin to happen. In this lesson, we're going to find out how enzymes can affect that activation energy required to allow chemical reactions to occur at a rate that's fast enough to sustain life. What we want you to focus on in this lesson is what role do enzymes play in living things and what kind of things are going to affect their function. A second point that we want you to think about is the fact that enzymes are going to speed up chemical reactions that take place in the cell. And the third point is that you're going to find out that temperature, pH, and regulatory molecules are going to affect the activity of enzymes. What exactly are these protein molecules called enzymes going to do to help keep us alive? Let's see if we can figure it out. Now, to begin with, we know that some chemical reactions are too slow, or else they have an activation energy that is too high in order to sustain life for us. So chemical reactions that are too slow are aided by the effect of molecules called catalyst. So a catalyst is a substance that speeds up the rate of a chemical reaction. And what it does is to lower the reaction's activation energy. To explain this concept, enzymes are proteins that act as biological catalysts, and what they're going to do for us is to speed up chemical reactions that have to take place in our cells. And they work by lowering the activation energies. And this is going to very dramatically increase the rate at which reactions are going to occur. Let's examine the graph, the effect of enzymes. If we look at our horizontal axis, our independent variable is the course of reaction, which basically means that we're thinking about time involved in this reaction. Our vertical axis, our dependent variable, is going to be the energy that is required to make this reaction happen. Focus first of all on the reactants and notice the energy level that the reactants have. Then look at the products. So we know that this is a chemical reaction that is going to give off energy. Now next we want to look at the blue line and the red line and compare. The red line represents this reaction pathway with an enzyme present. The blue line represents that reaction pathway without an enzyme present. And so what we can see is that the activation energy is reduced from this level to this level because we have an enzyme present. And so the overall result of this is that we're going to have this reaction occur much faster because it doesn't take as much activation energy to make it work. Let's return to our example of chemical reaction involving carbon dioxide and water in our blood. We know that carbon dioxide is produced as a result of respiration, and so the more you exercise, the more carbon dioxide you have produced, and your body needs to rid itself of this compound. And so in your blood, carbonic acid is going to be produced. And in this case, then, carbon dioxide plus water is going to form carbonic acid. This particular reaction is aided by the enzyme called carbonic anhydrase. This is going to change the rate of this reaction by a factor of 10 million. And so that makes this reaction occur very quickly, and it allows you to rid your body of the excess carbon dioxide, and therefore your blood is going to maintain its normal pH level. A characteristic of enzymes are that they are very, very specific, and what that means is that for every reaction, there's an enzyme that is going to be present that allows that reaction to occur at a rate that makes it useful to us as living things. Another feature about enzymes is that if you look at the name of them, the name is usually derived from the reaction that it catalyzes. So when you look at carbonic anhydrase, you know that the carbonic refers to the carbon dioxide. The anhydrase, anytime a word ends in ase, nearly always refers to an enzyme in living things. And in this case, it's called anhydrase because it is going to stimulate or aid the reverse reaction that removes water from the carbonic acid. Now, I wonder if you would know where that's happening. If you answered that this is going to happen in your lungs so that you can rid your body of carbon dioxide, give yourself a point. Now we're going to talk about the structure and functional relationship between an enzyme and the molecules on which it has an effect. So for a chemical reaction to take place, the reactants must collide with enough energy so that the existing bonds will be broken and the new bonds can be formed. Now, if the reactants do not have enough energy, then they're going to be unchanged after that happens. And so in this case, the enzymes are going to provide a site where the reactants can be brought together to react. And this site is going to reduce the energy needed for the reaction. Now we're going to explain the enzyme substrate complex. Looking at our illustration, first of all, note that the green part on this diagram represents the enzyme. So we're going to follow the action of the enzyme in this chemical reaction. The water and the carbon dioxide are the reactants in this chemical reaction. And so what happens here is that the water and the carbon dioxide enter that enzyme, bind to it, and because they are held there in close association, that's going to allow that chemical reaction to take place. In other words, we're going to find that the activation energy has been lowered because of their closeness to each other within the enzyme. The products of this reaction, as you can see here, the products of the reaction are the carbonic acid. That carbonic acid is then released from the enzyme. Now, the most useful thing about an enzyme is that it is used over and over again. So the fact that the enzyme catalyzes this reaction one time doesn't mean it can't do it again. And so that same enzyme is used over and over and over again. It does not become part of the product, as you can see when you look at the final product from this reaction. A bit more about the enzyme substrate complex. Substrates are going to bind to a certain site on the enzyme called the active site. Now this site is complementary to the shape of the substrate. And so this is called the activation site on the enzyme. And it is so specific that it is sometimes referred to as a lock and key mechanism. And generally the reason why there is only one enzyme for every chemical reaction is because the shape of the enzyme is fitted to the substrates that are involved in the chemical reaction that this enzyme is going to catalyze. Are there factors that regulate enzyme activity? Answer to that is yes. Some of those factors that are important are the temperature, the pH in the environment, and other regulatory enzymes. Thinking about temperature, if this is an enzyme that's working in the human body, it's going to work close to normal body temperature of 37 degrees Celsius. In conditions where it's too hot or too cold, that means that your enzymes are not going to work well and your chemical reactions are not going to proceed as quickly. Another factor that affects the chemical activity of enzymes would be the pH value. Some types of enzymes, such as the enzyme pepsin, which begins protein digestion in the stomach, is going to work best in acidic conditions. But if the pH in your stomach is altered by something like Pepto-Bismol, then that means that you are no longer going to have those enzymes work and you're not going to be able to digest your proteins as well. Another thing that affects enzyme activity would be certain molecules that are going to regulate these molecules are going to actually be switching these enzymes on or off as needed. We'll be studying more about factors that influence the enzyme activity in our virtual lab and our wet lab later in the week. Let's check your test readiness. Which of the following statements is true about catalysts? A. Do catalysts slow down the rate of chemical reactions? B. Are all catalysts enzymes? C. Are catalysts used up during a chemical reaction? D. Do catalysts lower the activation energy? If you chose answer D, you're ready for the test. Let's try another question. Enzymes affect the reactions in living cells by changing the, is it A, products of the reaction, B, the speed of the reaction, C, the temperature of the reaction, or D, the pH of the reaction? Be careful now. Think about exactly what it is that enzymes are going to do. Now, if you answered B, you're doing well. And let's go on to another question. There's a lot here on this slide to test your readiness for an exam. Let's look at the first question. Which enzyme would you expect to find in a bacterium growing in a hot spring? Well, on the graph, we can see that we're looking at temperature of the reaction as compared to the enzyme activity. And we have two different enzymes at work. Now, our question in this case has to do with a bacterium growing in a hot spring. So would you think that that would be enzyme X or enzyme Y? If you figured that it would be enzyme Y, you are correct. And the reason is that the activity of the enzyme is shown to be most active at temperatures that are at a higher level than enzyme X. Next, at what temperature do the two enzymes seem to have the same amount of activity? Well, the only place on the graph where the two lines intersect is this point. So at that point, which would be just about 50 degrees Celsius, it shows that our enzymes have the same amount of activity. Next question. What are the optimum temperatures for each enzyme? Taking a look at that, we're going to have to look at the highest point for each one of the lines on the graph. Enzyme X tops out at this level. That temperature would equate to about 40 degrees. Enzyme Y begins to show its greatest effect at about 60 degrees Celsius and continues over to around 90 degrees Celsius. Next question. Which enzyme is active over the largest temperature range? Well, in other words, which one of the enzymes has a broad area over which it's very active? And so our answer to that is going to be enzyme Y, because it shows to be active from about 60 degrees Celsius through around 90 degrees Celsius. Last question. Which enzyme would have the most activity in humans? What's our body temperature? Is our body temperature closer to 40, or is it closer to 60 to 90 degrees Celsius? Now remember, we're thinking Celsius here. So what's our answer? Answer is enzyme X. Remember these answers. You're going to see these questions again one day.